Thank you, Adam, for this wonderful introduction. My name is Laurent Bentonville. I'm the director of the Advanced Light Microscopy Spectroscopy here, and I'm going to do my best to tell you what we do, or at least a little bit of what we do in 5 minutes. But essentially what I would like you to remember is that using light and optical microscopy is very versatile. And we do imaging at all length scales. So from the nano to the micro scale, usually using fluorescence microscopy, so we can do and characterize single molecules all the way to tumors in small animals. And how do we do that? Essentially, most of what we do is fluorescence-based measurements. We do that at all length scales, and we are using 6 dimensions in our imaging. So the XYZ, that's your 3-dimensional imaging. And we can do that from nanometer to centimeters. Temporal, we can image from nanoseconds to days. And spectral, we can do it from the UV all the way to the infrared. So here are some examples to illustrate how we can actually mix and match all of these dimensions. Here is an example of 3D volumetric imaging. Most of the examples I'm going to use are taken from our biological samples, but we do a lot of nanomaterials as well. So here's another example of 3D imaging here combined with spectral, so multiple colors in one pass in 3 dimensions. Here we are looking at metastatic cells in green, actually covering a blood vessel in red, pretty deep into the brain of a mouse. So very powerful. You can slice it in every direction you want to really understand how this metastatic process develops over time. Here, we can use non-linear optics to probe even further deep into the tissue. Here, what's interesting is the pink signal is actually unlabeled structures into your sample. That was collagen. So we can do that by using a process called single harmonics generation. So here what I wanted to illustrate is the fact that we can image and stain samples through these kind of nonlinear processes. And here is a really gigantic slab of tissue that we can actually image in 3 dimensions, combining 2-photon microscopy and SHG. So 4 dimensions again. And here we can image really fast using 4 dimensions, X, Y, and Z, looking at a super fast volumetric imaging here of the beating heart of a live zebrafish. So from that kind of movie, you can understand that optical imaging is non-damaging, so we can image actually live samples in real time. All right, and another very good assets that we have is we have been one of the first labs here to introduce super resolution optical microscopy in order to close the gap between what a normal light microscope can do, which is limited by light diffraction. And this is what you will usually see if we want to relate the structure and the size of the structure we image with an electron microscopy image. So that would be your confocal microscope, about 200 nanometer resolution. Here we have here at CNSI, and we'll have demos if you come to the lab, STED microscopy, where you can push the limit to about like 20 nanometers. That will be your little structure here in that electron microscopy image. And we have storm imaging that can push even the limit even further down to around 10 nanometers or less. And that will be that little structure in blue. And here are some examples. Again, here, STED microscopy is very powerful because you keep all the advantages of confocal microscopy. You can image live samples, you can do it in 3D, and you can do it in multiple colors using your spectral capabilities. So, and you can, of course, apply that to nanomaterials and biological samples. Here is our team. That's our expertise. So please talk to us. We are here. Ellie here is in the back, and Brian is probably in the lab, but please talk to us. And really our expertise is not just the instrumentation, but our knowledge. We have a lot of experience. We can customize microscope for every experiment. We can assist you with sample prep. We consult to really design the entire pipeline of your imaging experiment. And we are also very well versed into data processing and visualization and customizing your experiments. So with that, it was really a bird view of what we do. So if you want to see more, please come downstairs and talk to us at any time. Thank you so much.